Assalamu alaikum everyone, welcome back to Regal Biology with Khadija. If you have ever wondered how researchers find gene sequence, scientific literature, genome or biotechnology resources online, then this video is for you. In today's video, we will explore the NCBI website from the very beginning, and I will explain what NCBI is, why it is one of the most important database in biotechnology, and how to use its different tools. Okay, so without wasting time, let's get started. Before we start exploring this individual NCBI database, it is very important to understand that this NCBI is just not the collection of separate databases, not these separate databases, but many beginners think same that it is just a collection of separate databases, but these nucleotide, clinical variations, proteins, biosamples, bioproject, these are all databases are interconnected with each other, and together form a very complete biological information network, you can say. So this connected system, this all connected system is called NCBI ecosystem. And this ecosystem works like a network where information from one database, for example, here gene, information from one database, one. Now, if we search this, suppose we have interest in this specific gene, and when we will explore this gene, what information will we get is not just about genes, but we can also get about the information about the description, location, is accession number, aliases, and here let's explore this DNA repair associated gene. It is present in humans. Look, the location of gene, the nucleotide sequence, the protein produced by this genes, protein coding gene or not, what is the description of this. You can explore many things just under one section. So one gene record becomes a gateway for many different biological information. Gene, if we want to check proteins and its transcripts, we can also check them. We can check the gene ID, length. Look. So if you are just getting information for one gene, you will not stop at the information of that specific genes, but also you can check the information of that specific genes genome, and you can check the command examples, data sets, each and everything. So this is interconnected system, and NCBI also connects the data with the publications. You can get information about the publications. For example, if thousands of researchers have studied this gene in cancer research, then this option here, PubMed, will help you uh, about the access of scientific information, scientific publications. Remember one very important thing, that NCBI is not just a group of separate databases, it is interconnected biological information network where every database is linked with other. And in the remaining video, we will explore each database one by one in a very short time, so that you will understand how to search, how to use these resources for biological research. Now let's start the job. First of all, if you just select all databases option here and search it, you will get information in this form. Your search all databases, your search is performed across all NCBI databases at the same time, instead of searching only one specific database. For example, if you search a gene name, NCBI will show related information from gene, nucleotide, genome, clinical, pubchem, proteins. This is useful when you are not sure which database contains information that you want to explore, or you and want to explore all available results in one search. Here, in just one search, you can get all information related to this gene in just one search. How to use all database options. Now let's explore all these databases one by one. First is assembly database. Suppose we search the assembly database. It actually is a database. It contains complete or partial assembled genome sequences of all organisms that are present and that are scientifically proven. So we will use it when we want to study an organism's assembled genome and chromosome information. Look here, you can check the modifier, the isolates. You can actually use this information to compare assembly of genes. Now let's study about bio collections. Here let's search it culture collection. Culture collection, and the information you will get here is about the biological collections such as culture collection specimens, and uh, it helps to identify where biological samples has taken and stored. So here you can see. Let's study bio project, and in bio project actually groups together all information that is generated from our research projects. Let's discuss about a virus. Mm, okay. If it helps you to understand the overall aim and data sets of the study. So here you can get information about this. Next is biosamples. Let's search the same virus again. Here in biosamples, it contains detailed information about biological samples used in experiments, including source, tissues, organisms, and collection details. So here you can get all information about samples that are used in the experiments. Let's search books database. It provides very free biomedical textbooks, manuals, reference books available through NCBI. They are free to open. You can learn from there free. Okay. Next, let's move toward next database, and our next database is clinical variations. It contains information about genetic variations and their relationships to human diseases. It is widely used in medical genetics and clinical research. Here you can see the gene, its type, its condition. Here you can get information all related to this. Let's study next database. The next database is conserved domains, and this database actually identifies conserved domains of proteins and the functional regions within the protein. Especially it helps to predict protein functions, so we can get information about the protein functions, conserved regions inside the protein. Here you can get information about that. We will discuss in detail about these all databases again in separate videos one by one, but here I'm just giving you an overall overview. So next is genotype and phenotype data. Here you can get genotype and phenotype data. In this database GAP, it stores information about genotype and phenotype. Data from studies investigating a relationship between genetic variations and human traits or diseases. Gene, the very useful and routinely used database is gene, and in gene database it provide information of about the detailed information about the gene, including their functions, including their locations, lysis, sequences. So if you want to study them, then select it and study it. Genome, let's study genomes. Genome database it actually provide the complete information about genome, information of thousands of organisms. Total 3.65 million genomes. Eukaryotes, archaea, bacteria, viruses. You can get all genomes here. Information of about all genomes. Next is GEO datasets. It stores complete information about gene expression. Mm, Gene expression experiments like defective viral genome from distinct genome regions. In this data base, you can get information of gene expression experiments generated using a microarrays or next generation sequencing technologies. Next database, GEO profile displays the expression level of a gene. Here you can check the expression level of a gene across different experiment, tissue, cell types, and disease conditions. It helps the researchers to understand when and where the gene is active and also how the expression changes under different biological conditions. So, this is the expression. You can check the expression from GEO profile. In different cell lines, it helps the researchers to understand when a gene you can say is active and how is the expression under different biological conditions. Database is GTR. Okay, it's a genetic testing registry. Let's study the gene, same gene, for example. Let's study it. Okay, here in genetic testing registry GTR, here you can it can provide information about genetic tests available for specific genes. For example, let's discuss this. It includes details about the laboratory offering the tests and the test methods, the conditions, and the conditions in which it is being processed, and the clinical purpose of this test is identical protein groups. Let's study some protein to cytochrome C protein. More specific. Okay. Next, it helps to researchers to understand that you can say um, to understand and to compare also. It helps the researchers to compare or conserve protein sequences under evolutionary relationships, like their functional similarities or not. Here you can check identical proteins, the FASTA sequence. Here you can check identical proteins. Okay, the next website is MedGen, like medical genetics. Suppose we search here a very common genetic diseases like what should we search? Let's search cystic fibrosis. Yeah. Okay. Search cystic fibrosis. This website organizes information about human medical conditions, diseases, and phenotypes. It connects the disease with related genes, genetic variations, clinical features, and other NCBI, you can say, resources such as gene, clinical variation, website, and PubMed website. Here you can get information about that. Here, look. It's a professional guideline, PubMed information, etiology information, all information you can add. Here you can also move to next information by related information getting through this site. Okay, next. MESH, here I'm going to study a disease. Let's study the same disease. Okay. Here you can study this disease and it actually provides standardized medical terms that are used instead of relying on different words used for researchers for the same concept. This MESH website assign a common heading that makes searching scientific information literature more accurate and efficient. You can see here, look, there are so many builder options here. NLM catalog. Let's study the molecular biology because this database provides you information about books. So let's study it. Okay, here you can study about molecular biology books In this catalog, it contains information about my biomedical books, journals, and other resources available in National Library. It includes details about author's name, publications, information, and subject category. Is about nucleotide. This is most commonly used web database also. Let's study gene. Suppose BRCA1, sorry. BRCA1. Okay. Okay, BRCA is a repair associated nucleotide database. Information that will provided by nucleotide database is store information about DNA, about RNA, and this information is submitted by different scientists around the world. Each record contain information about sequence along with the here you can see the sequence of nucleotide here. Okay, the sequence of nucleotide and also if you want to know the gene name, you can also get information from here. Here you can get the version, the locus, the DNA liner, uh, which chromosome number 17, its origin is Homo sapien. So you can get information from here about gene name, functions, reference. Database is about two main databases here, and they are all interconnected with each other. OMIM, it's an online Mendelian inheritance in men. Yes. Okay, let's study the same gene, BRCA1. It is very comprehensive database. It provides information about human genomes. Okay, and let's just verify that we are humans. Okay, here. No. Okay, look, this is very comprehensive database. Here, it provides information about human genes, genetic disorders. It explains how mutations in specific genes are associated with inherited disease, including their clinical features, inheritance pattern, and references. Here you can study all of this. Also, this is very comprehensive website. You can study phenotype, inheritance, line Mendelian inheritance in men. Next, our database is PMC. It's PubMed Central. Let's study um, any technique, CRISPR. Yeah, CRISPR gene editing. Here, you can this database will provide a digital archive of full text of biomedical research articles. Unlike PubMed, which where you can also get articles, here it mainly provides citations and abstracts. The main information, the citations and also the abstract. PubMed Central provide complete article, like the full article researchers can read, including the experiment methods, methodologies. Here, protein. Here you can get information about the name of the protein is hemoglobin. Hemoglobin. Okay. You can get information about protein function, protein structure, um, the literature about this protein, scientific literature. It actually helps the researchers to understand the protein and their biological roles. Here you can see. So, in cluster. Yes, protein clusters. In protein cluster database, let's search hemoglobin, and in this database, this database contains information about the evolutionary relationship. Look, hemoglobin has evolutionary relationship with this evolutionary relationship that is shared. It helps the researchers to compare protein from different organisms, identify conserved regions in different organisms, and study protein evolution and their functions. After protein cluster database, our next database is protein family models. Okay. Let's discuss about protein family. One protein family, protein kinase, suppose. Protein kinase. This database, here you can get information of protein family models. This database actually provide models about the protein families, the about their conserved regions, sequence patterns, and domains. Here, these models help researchers to identify unknown proteins and predict their functions. Suppose after getting information, you can predict the function of your unknown protein and understand the evolutionary relationship with other proteins that are present in other organisms. Here, CAM bioassay. These suppose you are performing a bioassay on aspirin medicine. Okay, aspirin. Actually, in this database, all the information about biological activity tests performed on chemical compounds will be present, and you can get information about that. It shows how compounds interact with biological targets, such as proteins or enzymes. Here you can study them. Look how it interact with other proteins, other enzymes. You can get information. Targets, bioassay annotations, same publication bioassays that has been performed. You can get information about this. Next is CAM bioassay, PubChem compound. Again, let's search aspirin. Here you can get information about the chemical structure of aspirin. Properties, chemical structure, chemical formula, identifiers of chemical compounds, primary hazards or not. Here you can get information about this. Database will provide you information about uh, aspirin, that is information provided by different contributors, including their description, their sources, their external ID, source category, sample information. You can get information all about this. And also you can know the synonyms, that is supplied synonyms, depositors. Let's study about CRISPR. Yes, CRISPR gene editing. Because in this database, um, it is a database of biomedical research articles, including the citations, abstracts, and scientific journals. It's a articles providing site, you can say. Study after PubMed is SNP, SNP. Okay, SNP. Let's study some kind of gene, like yeah, BRCA1. Okay. In this database, information about genetic variations, especially single nucleotide polymorphism, will be present. Variation type and location, frequency, and its association with different diseases. You can get here. Variant detail, clinical significance, submission, history, publications. You can get all information like which population, subgroups, reference allele. You can get information here. Okay, the next database that we are going to study after SNP is SRA. It's a sequence read archive. Mm, and let's study about the sequence read of any organism, suppose virus. SARS. Oh, actually, I'm using the same genes and the same organisms again and again for your analysis here so that you can study and understand them. Here, Okay, in this SRA database, it stores raw sequencing data generated from next generation sequencing, like who submitted external ID, including DNA and RNA sequence reads, all experiments. You can get information. Let's read archive SRA. Next is structure, taxonomy. Okay, in structure, let's discuss the structure of any protein. Suppose hemoglobin. Hemoglobin. Okay, let's discuss the structure. Here you can, in this database, it provides information about three-dimensional structure of protein, of biological molecules or proteins or nucleic acid. It's a three-dimensional structure. You can get information about three-dimensional structure here. Okay, full feature, it is 3D view. In this database, you can get information about the protein type. Look, this is all about protein, its details, its conserved domains, its clinical variations, its SNPs. You can get information about anything that you want, and you can also get details that in which region, which site, what is a change or not. After structure, um, taxonomy. Again, let's study the taxonomy of virus SARS-CoV-2, a very famous virus. Okay, let's study the taxonomy of this virus. In this database, organisms are organized according to their classification. It's the severe acute respiratory syndrome virus, including their domains, their kingdoms, their genus, species. You can get information about the whole biological organization of this organism here. Okay. Is after taxonomy is Toolkit. Study the in Toolkit the different tools like BLAST, sequence analysis, genome analysis. Let's study BLAST. Okay. This provides software tools. Here, programming resources. This is used to analyze biological data. The document here. So you can get information about software tools and programmings here. Next is toolkit. Toolkit all. Here let's search again blast. And here you can get information about NCBI software tools and resources again. Look. Here. Organism, size, type, target types, nodes, each and everything. Next, finally, this is our last database that we are going to study, Toolkit book. Molecular biology of cell. Right, you can get information or you can get books, textbooks, scientific documents, sequence features. You can get books here, freely available books here to study. This is all about databases. Now the question is how information is stored in NCBI. You might be wondered that NCBI contains millions of records, including DNA sequence, protein, and articles, genomes, disease-related data. Where does all this information came from? Who uploaded this information, and how does it become available on NCBI? The answer is NCBI does not create all biological information by itself. Most of the data is submitted by researchers, scientists, universities, and institutions like sequence centers from all over the world. For example, scientists perform a genome sequencing project. They generate a large amount of DNA sequence data. After analyzing and preparing their data, they submit it to NCBI database, such as nucleotide genome. Here they submitted like nucleotide genome, genome assembly, sequence read archive. Similarly, when researchers publish scientific studies, the researchers' information about their research articles become available through databases like PubMed and PubMed Central. Before adding data to databases, submitted information goes through a very checkpoints where the standardization process check that this information is correct, reliable, and useful or not. Now that we understand what NCBI is, my upcoming videos, I will explain each database one by one in separate video, like which database is it, which information it contains, why researchers use this database, how to use this database, how to understand the results of this database, how to download and use the available information. We will start with the most commonly used database such as gene, nucleotide, genome, protein, and assembly. We will discuss all important and commonly used databases in very detail. By the end of this series, you will have a complete understanding of how to navigate NCBI and use it as a powerful resource for biology, biotechnology, genetics, and research. So, in our upcoming video, we will explore the world of biological database with NCBI. If you are a student, researcher, or someone interested in biotechnology and bioinformatics, this series will help you to understand how to navigate and utilize these all resources step by step. So don't forget to subscribe and stay connected for the upcoming tutorials. Thank you for watching, and I will see you in our next video.